Last lecture, we have been talking about type of respiration, and we said that respiration can be costal or thoracic and abdominal. In a costal or thoracic respiration, we can see for women. Hello.
Hello,
And
sorry for being late.
okay, come. And the male type, this is abdominal respiration with using of diaphragm. And I haven't told you that abdominal respiration is more effective than thoracic one. When we are talking about investigation of respiratory function, we can use spirometry. Is an equipment used for measuring the volume of air inspired and expired by our lungs. During this method spirometry, this old one type on the photo, this new one with using of computer. And during spirometry, we can measure and can register next volumes. Tidal volume, this average amount of air inhaled or exhaled with each of breath under in condition of the rest. Also, we can measure with you inspiratory reserve volume. It's mean extra volume what we are able to inhale after the normal tidal volume inspiration. At the same time, we can measure expiratory reserve volume, this amount of air that can be forcefully exhaled after normal tidal volume expiration. Also, after expiratory reserve volume, anyway, in our lung, we have residual volume. This amount of air would remain in the lungs after a forced expiration. It means that how we will try too much, anyway, inside of our lung, we will save some volume of air, and this volume we never can expire from our lung. This residual volume started to be present with us in our lungs after our first breath. Also important to know about total dead space. Dead space, this some amount of volume which not involved in the process of gas exchanging. Total dead space consists of anatomical dead space and alveolar dead space. This volume named as physiological dead space, so it's mean normal amount of volume which not involved inside of process of gas exchanging. I will be back here. When we are talking about anatomical dead space, we are talking about respiratory pathways, because I told you that here not possible to have any gas exchange. Also, in normal condition, in case state of rest, top of our lungs also not involved in process of gas exchanging. Only when we have forced respiration or during physical activity, in the top of our lung increase blood circulation, and this time only we have starting of gas exchanging this area. Alveolar ventilation, main formula, what can we calculate with you? Minute ventilation, tidal volume, of average for adult person, it will be nearly 500 milliliters. multiply frequency of breathing. Normally, for again adult healthy person in state of rest, we have 12-16 respiratory movements during 1 minute. It means that frequency of our respiration will be 12-16 breaths per minute. Alveolar ventilation rate, we can calculate frequency again of respiration, but from tidal volume we should delete it, dead space, volume of dead space. As usually physiological dead space for adult person we calculate as 150 milliliter. It means that if 500 milliliter general tidal volume minus dead space, we will have like 350 milliliter, which included in the process of gas exchange for every respiratory movements, for every our process of inspiration. Gases exchange, how is going on? When oxygen moves to our alveoli, we calculate plus minus that partial pressure of oxygen should be nearly 100 millimeter Ag. The same time in our blood, of course, we have lower concentration partial pressure of oxygen. It will be nearly 40 millimeter Ag. About carbon dioxide in blood, because it should be happen exchange also for carbon dioxide, and carbon dioxide in the blood 46 millimeter Ag, but in alveolar 40 millimeter Ag. That's why we have with you difference between partial pressure inside alveoli and inside our blood. That's why this is main factor for gas diffusion. And important for you values is diffusing capacity of respiratory membrane. It's marked as DCRM. And for calculation DCRM, we should multiply surface to the what we have gas diffusion, multiply coefficient of diffusion for different gases. This is standard value, always K, this is constant value. And thickness of membrane. A thickness of membrane we calculate as L. And that's why diffusing capacity of respiratory membrane will depend on surface where happened diffusion, coefficient of diffusion, and thickness of membrane through the what happened gas diffusion. During physical exercises could be changed surface through which diffusion take place, because I told you right now that in the top of our lungs, in state of rest, we not have with you gas diffusion. But during physical exercise, it's happened also. That why surface for gas diffusion increase. And when we have active respiration, this time thickness of membrane decrease. More thin happened. And that's why as a result, we have with you changing of diffusion capacity. Ventilation perfusion ratio. Ventilation perfusion ratio acts on gas exchange, of course. And in state of rest, we have with you three zones. A zone of apex of our lung, zone 1. Here will be different. You can see here different in pressure in alveoli, in artery and vein. When alveolar pressure is increased or decreased, we will have changing of position of gas exchange. When happen gas exchange, we have with you transportation of gases, and how we have been talking before, we say that gas transport happened because of oxygen transportation. Oxygen could be transported by red blood cells, 98% of oxygen, but 3 milliliter of oxygen could be transported by 1 liter of blood plasma. So it means that it could be dissolved in plasma. And here also I remind you the value what is named as oxygen blood capacity. 1 gram of hemoglobin able to join 1.34 milliliter of oxygen. And here we have with you curve of dissociation, hemoglobin dissociation. In the lung, we have the less dissociation because not need. Oxygen should be not release this area to tissue. Hemoglobin full of oxygen. When happened transportation of hemoglobin by our blood, around tissue has less partial pressure of oxygen. That's why hemoglobin more quickly will dissociate it and will release oxygen to our tissue. And we say that effective dissociation happens when partial pressure will be less than 80. Partial pressure less than 40. Graph of dissociation, I told you that we have some situation when it's happened declining of this curve of dissociation to the right side or to the left side. If we are talking about declining to the right side, this time we are talking what does it mean to the right side? It means that hemoglobin dissociation happened more quickly, faster. This time, which factors can influence this process? Increasing of temperature, increasing partial pressure of carbon dioxide, decreasing of pH, so increasing acidity. And opposite story, when it declined to the left side, it's happened when decreasing of temperature, when we are frozen too much, when partial pressure of carbon dioxide decreased, and when pH will be increased, when we are talking about alkalosis. By the way, this situation when carbon dioxide can be decreased, when we are talking about artificial ventilation in a reanimation department, it's happened. And what does it mean declining to the left side? It means that dissociation, hemoglobin and oxygen happened not so easy, and it's so big problem for releasing oxygen to tissue. Carbon dioxide, when we are talking about transportation of carbon dioxide, this case we say that 7-10% dissolved in blood plasma, chemically bound to hemoglobin 20%, and bicarbonate ion in plasma 70% is transported as a bicarbonate. So most of carbon dioxide dissolved in blood plasma. When we are talking about respiration, regulation of respiration, here we have with you respiratory center. Respiratory center located in the medulla oblongata. There we have dorsal respiratory group and ventral respiratory group. Interesting that dorsal respiratory group consists of only inspiratory neurons. They responsible only for inspiration. Ventral respiratory group, it's like additional one group, and neurons which located there, they responsible for inspiration and for expiration. But I would like to remind you that last time I told you that normally in case of rest, we have with you active only inspiration and expiration happen passive. When we are talking about forced respiration or when we are talking about physical exercises, training, some emotional stress, this case will be added expiratory neurons function, and expiration also could be happen by active way. Upside medulla, we have a center which named as apnoistic center. Apnoistic center. Apnoistic center stimulate inspiration. Also, we have with you upside pneumotoxic center. Pneumotoxic center induce inhibition of inspiration. Because of pneumotoxic center able to inhibit inspiration, this center responsible for tidal volume and for frequency of our respiration. Important moment that all this center, and first of all dorsal and ventral respiratory group, they send signal to abdominal and intercostal muscles and to diaphragm through the spinal cord. Mechanism of regulation, main factors which has influence for our respiration. Let's look what we have here. Main factors, peripheral chemoreceptors and central chemoreceptors. Important moment. Again, I would like to underline for you that peripheral chemoreceptors, they able to activate it by oxygen concentration decreasing, by carbon dioxide increasing, and H+ increasing. Central chemoreceptors, which located in the respiratory center, they are sensitive only to carbon dioxide and H+ ions, but they are non-sensitive to oxygen. This important moment, please pay attention for that. Also, for activation of our respiratory center, we have as activations and inhibition by pain or by emotional stimuli acting through the hypothalamus. Also, by nervus vagus through the activation of stretch receptor in lungs. Stretch receptor in lungs activated when we are beginning inspiration. These receptors are stimulated and happen Gehring-Breyer reflex. Gehring-Breyer reflex. And inspiration is stopped. For adult person, this is not important reflex, but for newborn baby, this important reflex because their respiratory movements is not so good regulated. That's why this reflex is important for will be not damaging, not happen damaging of lungs. They doing inspire, happened irritation from stretching receptors and beginning expire. Also, we have with you irritant receptors. Irritant receptors we have inside passageways. Irritant receptor could be irritant under some dry air or some particles can come in our respiratory passageway, these receptors will be excited, and as a result, we will have with you defensive reflex as coughing. Normally, it's happened coughing. One more important factor which influence for our respiration, these receptors in muscles and joints. When we begin physical exercises, our muscles, our joints also sending signal to our respiratory center, and we have stimulation of respiratory process. Also, one more important moment, that our brain cortex also able to control our respiratory center. But how is it happened? Actually, I say wrong, because our cortex is not able to control our respiratory center, but able to control our respiration. How is it happen? We cannot control respiratory center, but we can control our respiratory muscles. And when we, for example, making with you deep inhale or deep exhale, we can control it from our cortex, but we control only muscles. If we will stop with you our respiratory movements, if we will keep our breathing and will stop breathing, after 1-2 minutes, you're not able to continue this process, because a main for our respiratory regulation is respiratory center, and only respiratory center can regulate our respiration and movement of our respiratory muscles. That's why if you will decide to stop your respiration, will happen without any help, like extra help from outside. After 30, 40, 50 seconds, your respiration will be renewed. You will do it, this experiment, for your practical lessons. Respiratory adaptation to high altitude. When we are talking about high altitude, like high mountain, this time important to say that there is low partial pressure of oxygen. That's why the main problem, this is that why our body should be adapted for this condition. And we have 3 stages of adaptation to low partial pressure of oxygen. First, this immediate reaction. For immediate reaction, we have activation of our peripheral receptors, peripheral chemoreceptors, this one, which are able to indicate a level of oxygen. And as a result, we will have with you hyperventilation. But long-time hyperventilation decrease level of carbon dioxide, and it induce respiratory alkalosis. This increase affinity of hemoglobin for oxygen, and as a result, better oxygen loading in the lung. Second step, it's happened adaptation during few days. During few days, in our red blood cells, increase it 2,3 dpg. As a result, increasing 2,3 dpg, affinity of hemoglobin for oxygen decrease, and oxygen uploading to tissue is increased. During 4 or 5 days and 2 weeks, start to work our kidneys. Kidneys, for deficiency of oxygen, begin to create erythropoietin. Erythropoietin, this substance which stimulate for us production of erythrocytes. Erythropoietin has influence on red bone marrow. As a result, we have increasing production of RBC, and as a result, count of RBC and hemoglobin is increased, and oxygen content of blood also increased. decrease. If we are talking about respiration underwater, what happened there? Problem with respiration underwater is the next, that under pressure, gas solubility is increased. And problem is not that gas solubility is increased, but by the way, also sometimes happen problem and here because some gases could be toxic for us, which are not so high solubility in normal condition. And some gases can influence for our brain, and this is problem for divers, for example. Not trained divers never can go for deep area without some professional people, because that time can be changed gas solubility, and as a result sometimes happen that happen toxic effect, and they like losing their conscience. They not understand what is going on with them. They begin feeling funny, high excitation, and they can take out their oxygen mask and could be die. That's why only trained people could be go for area with high compression. Why in the picture you could see cola? Because you know that if we will looking on the bottle with cola in shopping, you will never see there any gas, right? But when we open cola bottle, this time we see so many gases. Why it's happened? Because water closed under pressure, and this gas what have inside bottle, it's soluble, dissolved in the our drink in cola. But when pressure changes, when pressure is decreased, because we open bottle, gas solubility changes, and you see so many bubbles in the bottle. Why I'm talking now about it? Because problem happened during process of decompression. What does it mean? When person go to the deep, and after moves to the up from the bottom of some area, happen reaction decompression. Same reaction how happened in the bottle of cola. If so high speed we will move up, it will induce creation of gas bubbles in the human body, like champagne blood. This is dangerous because this bubble will induce embolia, gas embolia, and will be stop blood circulation. Person could be die. That's why when we are talking about divers, you should know that if, for example, all time of diving, I don't know, for example, 1 hour or 40 minutes, only 20% of this time they use it for diving to the deep, and all 80% of the time they use it for moves to the up. For not so quickly happen change of pressure of the gas, and that time will not happen in high quantity of these bubbles. And one more important question, what should you know? This regulation of first inspiration of a newborn baby. When baby is birth after labor, what is happened? First of all, of course, its lung doesn't work. That's why for starting this first inspire should be happen some combination of some process. First of all, this is hypercapnia. Hypercapnia, it means that increasing level of carbon dioxide in the blood. This is this important factor for stimulation of respiration. Next one, this increasing of hydrogen ions concentration. Next one, this hypoxia and irritation of peripheral chemoreceptors because not enough oxygen. Next one, and not less than more important than previous factors. This we are talking about irritation of skin mechanoreceptors, nociceptors, vestibular receptors of other sensory system. After labor, baby came to absolutely different world with touching, with pain, with temperature, with light, with decreasing temperature down. And it's important for stimulation of first inspiration. Interesting that decreasing of skin temperature increased lungs ventilation on 30%. This is one of the important mechanism of respiratory center stimulation. Maybe you know that some people trying to birth not in hospital condition, try to birth baby in like private condition and birth in the water or something else what they are created. This is so dangerous because this time if they try to say we are trying to create more comfortable condition for babies, this is not normal. Why is not normal? Because for start first inspiration should be not comfortable condition, should be absolutely different conditions that baby stay inside of mother body. And one more important mechanism, this liberation of passageway from liquid and taking off the diver reflex. It's also interesting reflex. Even we say that have diver receptors. What is it, diver receptors? These some special receptors which combined like for tactile receptors and maybe a little bit baroreceptors and little bit thermoreceptors the same time, because they should make a reaction for the water. For water, we not have any receptors in our body. And this diver reflex created from diver receptors. Diver receptors located under our nose, this area, and inside our nose. Interesting that when we have irritation, not we have, newborn baby have irritation this receptor by liquid, it stopped first inspiration, like stopped breathing. And by the way, you also know this reflex when you washing your face or when water will come in your face, automatically you not control this process, automatically you stop your inspiring all time. When example, when you clear your face in morning, for example, you clear face and automatically thinking you stop your inspiration. You even not control this automatically happen. And for us, this reflex is not so valid and important, but for newborn baby, absolutely important reflex. And if you know, now it's also popular, this is swimming baby before they beginning walking or even yeah, you know. And
Yes.
and they teach babies for diving. How they are doing that, how you can explain for 2-3 months babies that you should stop your inspiration. It's not possible. But when baby moves down underwater, automatically it stops its breathing. And it's so easy if start this education from this time. So easy, baby will be learn how to swimming, and they are so easy, so good swimming underwater. And actually, my opinion, this all very healthy idea for baby because it's good for development and growing of muscles, joints, physical activity, for increasing development of brain. But at the same time, we should calculate other factor. We should be sure about coach who will train our baby. We should be sure about hygiene, because they're so important for not catching any infection inside this water. So this dangerous, these factors, but for general, for babies, this swimming is absolutely good and healthy.
Okay.
What else? Few moments about temperature. I will tell you. When we are talking about temperature, we have with you inside our hypothalamus spatial center. It's a center named as set point. Set point equal to 37.1. And this set point all time control what is going on with our temperature of our blood, of course. If temperature of the blood is increase, we have heat losing process, like increasing skin blood flow, evaporation, and some behavior change, and so on. If, for example, opposite happens, this temperature of the blood decrease less than 37.1, this time our set point understands that it's not enough and should be heat production of our body, and happen some behavior changing. We try to cover if it's possible, we try to move. If not control process, it's increased metabolic process, and also happen shivering, not control it, trembling of our muscles. Because of this trembling, we will have also increasing of heat production. Okay. Next question. Next question. Let's do 5-minute break, and I will open a new topic.
Okay.
Okay.
When we are talking with you about physiology of digestion, we should say with you the digestive system consists of a long hollow muscular tube or a canal or tract, which called as gastrointestinal tract or GIT. Also here we have with you accessory glands, including a salivary gland, liver, gallbladder, and pancreas. When we are talking about this GIT starts from oral cavity. After we have with you esophagus moves to the stomach. From stomach we move to small intestine, and after large intestine, and that is all. During our digestion, we have proceeding of all main process, like transformation from our food, what we are consist, to some nutrition like amino acids and carbohydrates, and so on. Four major activities of GI tract: this motility, secretion of juices and saliva, digestion and absorption. Digestion, under a term of digestion, we mean food broken down into absorbable molecules. Absorption, here we are talking about absorption of nutrients, electrolytes, water, and transportation, all this to the bloodstream. Ingestion of food is simply voluntary taking food into the digestive tract through the oral cavity. In our oral cavity, we have with you chewing mechanical influence on our food, chemical influence from our saliva. Here we have with you a receptor, taste receptor on our tongue. Taste helps to us, first of all, helps to us to identify toxins, maintain nutrition, regulate our appetite, immune response, and also has influence for gastrointestinal motility. And even from our oral cavity, we can induce not only motility, but secretion of gastric juice, working of our liver and our pancreas. Interesting moment, taste on our tongue. Normally, you should know 4 main tastes: sweet, salty, sour, and bitter. Sweet, more sensitive for a sweet, we have on the apex of our tongue. Salty, yellow area, sour or acid on the lateral part of our tongue, and bitter taste like on the base of our tongue. But like 20, maybe 30 years ago, scientists discovered new one taste, and this taste is named as umami. Did you hear about it? Umami.
Yes.
Here. What does it mean, umami?
I told if you're having noodles, you know, these Chinese noodles, it's like taste when you taste it, and it's called a new taste,
Yeah.
and it's called umami. Yeah.
Yeah, it's all people thinking that it's so tasty, yes, and they added this umami. But what is it, umami? In our products, it's named as sodium glutamate, or substance which increase taste sensitivity. Why it's increased taste sensitivity? Because you see, for this umami receptors, we have so many receptors for this taste, like all our tongue filled by these receptors, all our tongue. What does it mean, umami? They name it umami, but normally these receptors for glutamate. Where and when did we hear with you about glutamate? What is it, glutamate? Did you hear before glutamate? I mean, inside our body.
Yes, yes. I
Yeah.
heard it in the excitation. No, not excitation. Inhibition.
Excitation.
Excitation. Okay. Yeah. Glutamate.
Excitation neurotransmitter.
Yes.
This substance which created by our brain and induced transmission excitatory signal. What is interesting, so it means that this substance can be produced by our own body. That's why when we have excitation of all these receptors, and when glutamate moves to our body, to our blood, it's able to induce excitation, everything, everywhere where moves this glutamate. And first of all, it moves to our stomach and our intestine. That's why people who use so much food with glutamate or with umami, they as usually have problem with their stomach. And as usually, stomach has inflammation because so much excitation as a result will induce inflammation in this area. When we are talking about using glutamate in our food, as usually it could be some Chinese food, obligatory, of course, plus it could be different spicy, it could be chips. There's so high level of glutamate, chips and different salty snacks as usually consist of glutamate because it seems like very tasty, but tasty only because of presence of glutamate. If we are talking about natural products, also they consist of glutamate inside tomato, inside mushroom, and inside some kind of cabbage. I don't remember, like Brussels cabbage and something else. Also consist of glutamate. That's why people feeling like it's tasty. What is important about glutamate in our daily meal? If we are talking about, for example, some prepared meat or sausage, there also high concentration of glutamate. What I want to say you is that, for example, if you will make this experiment, but better not do that. If for small children, doesn't matter, 2, 3, 4 years old, you will start to give products with glutamate, like some snacks or some sausage. After, if you will give normal food, healthy food, they will not agree to eating. Because why? You see, normal healthy food, which receptors irritate, and umami, which receptors irritate. It means that for small children, it's normal. They not understand what is good, what is bad, but they so good understand what is tasty, what is not tasty. And this is a huge problem because after, for example, normal meat, they don't want to eating, they want eating only sausage, or they want eating something tasty like that. It's not good. Not good for what? For, first of all, it's not good for their stomach. This is not good for their intestine. But interesting, I have read about some investigation. They say that children who using these products with umami, small children, their behavior also will be changed. Because this excitatory neurotransmitter, and for small children, this excitatory neurotransmitter able moves to the nervous system, they say, and children behavior also change. They are beginning to be more hyperactive, more nervous, more irritable for different irritants. That's why for small children, it's really is not healthy food. Our tongue has innervation by 7 and 9 cranial nerve. 7 and 9. Also, pharynx innervated by 10 cranial nerve. And all this innervation included in the process of swallowing. A little bit talking about oral cavity and what is going there with saliva. Chemical process. Because of saliva present, we have with you chemical digestion. Chemical digestion happens for starch or for carbohydrates in the oral cavity, because there we have with you enzymes which called as amylase. And you know that amylase responsible for breaking down carbohydrates. That's why if you will take some carbohydrate food, I don't know what, for example, some pasta or a piece of bread, but not sweet bread, and you will chewing a long time, like 1 minute, you will feeling sweet taste because will be happen breaking down of carbohydrate to glucose. Also, if we are talking about saliva production, we have a huge volume of saliva per day, like 1.5 liter per day. Is a slightly acid solution of salts and organic substances secreted mainly by salivary glands. Main salivary glands, here we have two parotid glands, sublingual gland and submandibular gland is shaped as U shape. Basic saliva components is the main components. It's water, of course, also iron, sodium, potassium, calcium, chlorine, hydrocarbonate or carbohydrate. Proline, rich proteins for protection of this animal, enzymes, amylase, and also we have here lingual lipase. Actually, they cannot explain for what do we need lipase in our oral cavity. Maybe we're thinking maybe for some protection from piece of food which stick between our teeth.
Okay.
Immunoglobulins A for protection, here releasing mucin. Mucin is glycoprotein for lubrication of food and protection of oral mucosa. Lysozyme and lactoferrin. Lysozyme, this is so high level protection from bacteria. And normally pH of saliva is about 7. Important moment that 1 second. Important moments that when we are talking about health of our teeth, this time important to why is important to clear our teeth? Explain me, please. Why is it dangerous to not do this hygiene of our oral cavity?
Yes, because the pH will be actually will go down, and when the pH goes down, it could lead to the formation of caries, so caries lesions, and that is why we should actually protect the teeth from the plaque.
Absolutely. Absolutely right. But what is the factor which will induce decreasing pH?
Decreasing pH, pH, plaque, you mean?
No, pH level of pH.
Sorry.
Why is
pH, what could be actually if we take some food, yeah, every time if we take some food, it will be decreased. If we have some special food, for example, lemon, yeah, it could decrease.
Acid food, okay, but if not acid food, why say that, for
Yes,
example,
but
sweets is so dangerous for our teeth and it's induce caries?
actually, because the bacteria will digest and it will turn into glucose, and this will be bad for the enamel because it's
Not
consisting
glucose,
actually.
dear, no, not glucose, but bacteria. You're absolutely right.
Yes.
Bacteria during their life cycle, when they're eating, they normal breaking down. They very like everything what we are eating also. And
Yes.
when during their life cycle, they begin to release acid.
Acid.
And this acid, if we not clear our teeth, this acid will be destroy our animal and will be induce caries. Okay, great. Next, non-digestive function of saliva, this cleansing, how we say, protection, because there we have also leukocytes and enzyme lysozyme, and antibodies act against some bacteria. Also functions this moistening and lubricating of our oral cavity from some dry food, what we are catching in our oral cavity, and excretory function, because many organic substance, also urea and sugar and inorganic substance can be excreted in saliva. Regulation of salivation. Our salivatory gland innervated by sympathetic and parasympathetic system, both. But it's usually when we say about sympathetic and parasympathetic system, we say they are working like opposite to each other. But only one where they working like together, this our salivary gland. For salivary gland, and sympathetic and parasympathetic system will induce stimulation of salivation, but different chemical composition of saliva. When we are talking about innervation of parasympathetic system, by the way, you can see that by different nerve it happens, this innervation of a different gland. When we are talking about parasympathetic system, this system of digestion, that why when we have with you activation of digestion this time, we say that we need like stimulate of digestion. If we are talking about stimulation of all process of digestion, for our saliva should be so much watery. That's why we have increasing production watery saliva. It's important for this function, like moistening of our food, But when we are talking about sympathetic system, here is happened opposite story. Which opposite? Also happened stimulation of salivatory gland, but here without so high quantity of water. Here increased quantity of enzymes, immunoglobulins, because sympathetic system, this system of the stress. And you know very good that when we are nervous or when during physical activity, we are feeling that our mouth is dry. But it's not because we not have production, but because watery of saliva changed and water decreased, but enzymes and lysozyme is increased because our oral cavity preparing for protection. This picture for innervation of our tongue and connection between tongue and stimulation of our salivatory gland by parasympathetic system, because when something come upside our tongue for food, like begin releasing of saliva from taste receptors, signal moves to medulla, from there come by 9 cranial nerve and induce stimulation of our salivation. Mechanical process in oral cavity. Here we are talking about mastication of the food, the teeth cut and grinded and into a bolus. Because food we name as food when it's on the plate. When it's come to our oral cavity, we chewing and it's named as bolus. When we are talking about mastication, it's possible as voluntary and partly reflexive. So we not all time control our mastication, our chewing. Of course, usually it's happened by a reflective way. What else? Significance of our chewing and mastication, because here important for breaking down our food into small pieces for easily swallow it. Also expose food to salivary amylase enzyme for starting digestion of carbohydrates like starch, and help digestion of all types of food, especially cellulose containing food and vegetables. Swallowing. We have with you three steps of swallowing or three phases of swallowing. First one, this buccal phase, food pushed on them back into pharynx from the mouth. Pay attention that this time soft palate closed our respiratory way to the up, and our glottis should be closed now, enter to our low respiratory passageway. Step 2, epiglottis closed, you see, epiglottis closed, and that's why our bolus can easily move by esophagus to the down. And a third one step, when happens swallowing and again epiglottis is opened and our respiratory renew. Esophagus. Esophagus, this is a long muscular tube, like 25 centimeters. Interesting that esophagus transportation of our bodies, our food, happened by active way. Most of people imagine this process like, I don't know, like rubbish, some tube or something like that, like non-active process, but it's wrong because transportation by esophagus, this is active process. It's happened, you see, when we have some part of bolus, it happened like special wave, have constriction of esophageal sphincter, increased pressure, and happened like wave, less more tone. That's why our food transported to the stomach by active way. This absolutely important moment. On the enter to esophagus, we have esophageal sphincter, and on the enter to the stomach, also we have cardial sphincter, which close in normal condition. It should be all time closed for not giving to moves away food from the gastrum, from the stomach. So by the way, what I want to say is that if normally, theoretically, if we will eating in position vertical sitting, or if we will standing, or if we will laying, or even if we will move our head to the down, our esophagus anyway should be working in one direction from our oral cavity to stomach. Motility of stomach. Here we have cardial sphincter, here we have pyloric sphincter. Fundus body of stomach. Normally, all our food should be moved by this direction. For that, of course, we have as help motility of contractility and motility of our stomach. Different way of motility. You can see like food will be mixed here, and step by step through the pyloric sphincter will be released to duodenum. In our stomach, we have special secretory cells. Secretory cells named as parietal cells, chief cells, and mucus cells. By the way, I would like to ask you, for what do we need with your stomach? What is the function of the stomach? Normally.
Yeah, I would say in the stomach there will be also some components by molecule digested. For example, amino acids are digested in the stomach. Also, I would say fats or carbohydrates, which are further digested in the stomach.
Nick, you agree with Susan? Nick sleeping, I guess. No, dear, it's wrong.
Okay.
It's usually so many people thinking as you say. That stomach we need for digestion of proteins and carbohydrates and lipids. But so important to know that in the stomach we not have digestion, not lipids, not carbohydrates. We not have their enzymes for breaking down these substances. In the stomach happen only breaking down of proteins, only proteins, level of peptides, even not to level of amino acids. Amino acids will be happen in our intestine. Stomach only one function, this breaking down of proteins. For us, if we will compare with you digestion of proteins, lipids, carbohydrates, the most complicated process is digestion of proteins. This process needs so much time, so much energy and sources. And stomach working for us as storage because we are eating all food, yes, all meal, and it's happened as working as storage. But if we are talking about breaking down of substance, only proteins starting breaking down here.
Okay.
Also important moment, if we are talking about absorption, in the stomach also absorption is not possible. In stomach possible absolutely small quantity absorption of water and alcohol. That is all. More something is not able to be absorbed in the stomach. All absorption happen in intestine. Why is important? Why I pay your attention for that? For example, when we using some treatment, some pills, and these pills came to our stomach, it should be moves more in intestine. Why when we using some pills, time of action these pills is so prolonged? Because not possible to absorb it in the stomach. Most of pills are not absorbed in the stomach. They should move more long way. That's why time of absorption and action of pills take as minimum 30-40 minutes. Also, when we are eating with you carbohydrate food or fat food, if only carbohydrate, for example, we are eating, this carbohydrate not need to stay in the stomach. What they will do there? They will quickly induce evacuation and will move to the intestine because not have any process in stomach for them. But if we are eating meat or fish or some other variant of protein, this time they should be stay in the stomach. And then more protein we're eating, then more long time this food will be stay inside of our stomach. In stomach, how I told you, have three main type of secretory cell. Parietal cell and chief cell, main one. Look, chief cell release pepsinogen. Pepsinogen, this non-active form of enzyme. Pepsinogen, after activation to pepsin, able to induce digestion of proteins. On the picture, you also can see that here we have gastric lipase, but gastric lipase working only for newborn and small children for digestion of the milk. After this enzyme is closed and for adult person is not working. But pepsinogen converted to pepsin, it should be working all our life. Important moment, all of us, we know so good that in the stomach we have so high acidity. We have so high acidity because there we have is you releasing of gastric acid. Yeah?
Yes.
Gastric acid. Why do we need gastric acid? Gastric acid we need for activation of pepsinogen and conversion to pepsin. If in our stomach will be not enough acidity, this time pepsinogen will be not converted to pepsin and digestion of protein will be not possible. That's why you can so often to hear from people, oh, I have problem with digestion, I have problem with acidity, my acidity is high, and like that. Yeah, this problem because it can induce inflammation in the gastrum. But how do you think, what will be happen with people for whom acidity is decreased, not enough acidity? What will be happened?
Probably the pepsinogen cannot be activated or very low.
Yes, and what will be happened?
We have we have very low digestion of proteins, probably even very low, probably not.
Yeah, but what is important? Important is the next. If protein will be not breaking down, of course, we will not receive amino acid. This is first problem. But another problem that normally if protein will be not breaking down in time, they're converted to very toxic substances and will be happen process of decaying of these proteins, and as a result toxic substances will move to the blood. People feeling this time so bad, they feeling like intoxication inside their body. And you could hear that people say, you know, I cannot use meat or I cannot use any proteins, animal proteins. I'm feeling so bad after that.
Okay.
And all time I say, if you hear this, you should say this person, please go to gastroenterologist. The high chance that you have problem with gastric acid and with digestion of proteins. That's why all this happened. Also, parietal cell, they release intrinsic factor. Intrinsic factor is absolutely obligatory component for absorption of vitamin B12. And people who have low acidity, as usually it's happened because not enough stimulation of parietal cell, they also have low intrinsic factor. And as usually we can see in their blood anemia.
Okay.
So interesting, you see. So we came from problem with digestion and turned to the problem with the blood. And this have connection, directly connection through the parietal cell. Also, we have mucous cell. Mucus, they release mucus and bicarbonate as this important cell because mucus protect our gastrum from them. Our own acidity, yeah, from our own acidity. And this is important because if we will not have this protection, this time will happen our own digestion. So it will be working like our own gastric acid can damage it, and together with pepsin can damage it, our gastrum walls. Also, here we have for regulation next cells. Interachromophin like cell, they release histamine. Histamine stimulate gastric acid secretion. D cells release somatostatin, induce inhibition of gastric acid secretion, and G cells also releasing gastrin, and gastrin also stimulate gastric acid secretion. Okay. Acid. Acid help for activating of pepsinogen transformation to pepsin, provides optimum pH for action of pepsins, denaturate proteins, induce denaturation of proteins, kills bacteria in food like antibacterial action, help iron and calcium absorption. So if will be not enough gastric acid, also will be problem with absorption of iron, and again anemia, but already iron deficiency anemia. Promotes pancreatic, small intestinal, and bile secretion. Here you can see experiment, gastric acid and some piece of meat, not happen digestion, only pepsin without gastric acid, and pepsin plus gastric acid, you see that happened like full digestion. But full digestion not happened, of course, because here we have, how I told you before, dividing only till level of peptide. To amino acid dividing happen in our intestine, Okay, and also important to know that a secretion of gastric juice production happened also in 3 phases. First phase, this cephalic phase through the vagus. What does it mean, a cephalic phase? Gastric juice production happened when we see food, when we feeling smell of food, when we begin chewing food, when we even imagine we are hungry, if we imagine food, this time we have stimulation of gastric juice production. Also, second phase, this gastric phase, this main phase, when food moves to our stomach, here happened local nervous reflexes and vagal reflexes and gastric histamine stimulation, and we have main stimulation of gastric juice production, and also a little bit like 10% production of gastric juice happened during intestinal phase, when food moves to the intestine, and this time we have nervous and humoral mechanism of regulation in gastric juice production. Так, okay. I think this topic we will continue next lecture. For today, it's enough. Thank you for your attention.
Thank you very much. I have a question. This is organizational. I don't know if you want to record or we will stop. It's about when will